started, ma'am.
Yeah. Good morning, everyone. I welcome you all for the last lecture of our WINR and DST collaboration series on research methodology. So our topic for today is two types of study designs and how to remove bias from your study. Our speaker for today is Ms. Pooja Rawat. She's working as a clinical research associate in LabCorp drug development in Delhi. So over to Ms. Pooja. Please start your presentation.
Yeah, hi everyone. My name is Pooja and good morning to all of you. So let me tell you a brief of myself, then we'll start. So I've started my journey in clinical research industry in 2016, and in last 7 years, I worked with various firms, including Excel Life Sciences, Oxygen Medical, Lambda Therapeutic, LabCorp, Sanofi, with the various areas of expertise like ophthalmology, pulmonology, orthopedic, COVID-19, and the rare disease. So during this, I have been working with various kinds of study design and with the various kinds of clinical operations activities. So for today, I thank everyone for inviting me to this panel, and a special thanks to Dr. Mohita for giving me this wonderful opportunity to share my views and knowledge on this program of WINR. And I also thanks Dr. Nisha for her coordination. Last but not least, thanks to the all wonderful people who are who work in this course to make it successful. Okay. So I'll share my screen. Okay, Dr. Nisha, just confirm me that whether it has been visible or not.
Yeah, it's visible.
And now this.
Yes.
Can you see the PPT or anything else?
Yeah, I can see only the PPT.
Okay, fine. So my topic is types of study design and how to reduce biasness. So these are the following contents to which I'll be taking you, all of you, through these, like what are the types of research, what are the clinical phases. Basically, this is the hot topic after COVID. Most of the people who are, I mean, the layman one also knows about clinical research. So what are the different study design of the clinical research we are doing the research, and how we should reduce the biasness, and what are the methods by which we can reduce these biasness in the study. So you can see this is a broad spectrum of medical research. So the medical research is classified into a primary and secondary research, and the clinical and the experimental studies are performed in primary research, whereas the secondary research consolidate the available study as a review or a systematic review and a meta-analysis. These main area in primary research are basically medical research, clinical research, or epidemiological studies. So basic research include fundamental research. In almost all the study, at least one independent variable is varied, whereas the affected the independent variable are investigated. So the clinical studies include observational studies, intervention, and there are further classifications as accordingly. So basic research, which you can see here, the basic research is further classified into theoretical and applied, where some inception is there, or you can do some kind of testing. You are getting some raw data, the baseline kind of data, like through genetic studies or cell studies you're getting, or some kind of X-ray imaging kind of data you gather, and then you got to know that the dimension of that particular, you can say the enzyme or the cell, or how the substrate or the enzyme is binding to that particular. So these are the basic kind of research which further lead to the subsequent research, especially the clinical research. So once you'll get it, then you move especially to the clinical research, because it has a various phases. So clinical here means you are inducing that particular active ingredients to the human being. So prior to that, we should have a basic research. So I'll take you through the clinical one, which has been further divided into the experimental and the observational kind of study. And there are other parts of study which I'll not talk, because these are the secondary kind. So we are mainly focused on the clinical trial, because here we need to talk about the various designs and how we have to reduce the biasness. So through that, we are, begin with this clinical trial. So clinical trial, what it is? So it is a studies performed in human that are aimed at evaluating medical, surgical, or behavioral intervention, or you can say the treatment. Intervention is especially for the medical device. Treatment is for the drugs. They are primarily they are the primary way that researchers find out in a new treatment, like a new drug, new medical device, or it is a safe or effective, or the performance of the device is okay or not. So there are the various phases like phase 1, phase 2, phase 3, and phase 4. So you can see in the depicted what you can say, the diagram. Here the phase 1, before phase 1 there is a phase 0 study also, or you can say preclinical studies, and that I haven't talked about it, so I'll just a little bit inform you. So preclinical studies are basically done on animals, just to see the whether this drug is safe or not before introducing it to the humans. And apart from this, various cell related activities is being also observed, and that's just to check that there shouldn't be any cytotoxicity inside the cell, or leading it to the further change in other enzyme or other indications within the body. So preclinical trials are those. And usually in India, it is banned because of animal cruelty. So US, Canada, Japan, such countries are performing this. Somehow in India, very few of the organization, very few, 1 or 2 are doing this, but they are collaborating with one or another medical colleges. That's why they are getting this approval. Rest in a commercial purpose, it has been not approved in India. So let's talk about phase 1. So here you can see in the picture that 2200 healthy volunteers we are taking, not the diseased one, the healthy. I'll tell you why later on. And then in the phase 2, the sample size increased to 3200. You can see in phase 1, we are just checking the dose, whether and the safety, whether it is safe or not. And in the second phase, we are confirming the efficacy of that drug, that it is whether it is effective or not. And in phase 3, this is a confirmatory kind of phase 3, where we are confirming both the safety and the efficacy. And then comes to the regulatory submission. After all this data collection, we need to submit it to the regulatory bodies within the respective countries. They will check the data, and based upon the data and its outcome, they will give approval to the subsequent drug or to the device. And after that, we have to perform another phase of the study, that is called phase 4. That means post-marketing approval kind of studies. So this is what you can say a traditional method of doing clinical trials. And before that, there are various other methods also, like in new scenario, the computer-based drug designing is also very much popular. In this, we are just based upon the basic research, which I earlier talked about. Based upon the basic research, we are just checking the compatibility of that particular active substance with the enzyme, you can say, or with that gene, whether it is being compatible to inhibit the further process. So that completely comes under those diseases which are very rare and lead by genetic defaults, right? So currently we will talk about the traditional one first. Okay, this is the phase 1, where this is also known as non-therapeutic phase also, because here we are not bothered about the whether our active ingredient, whatever the XYZ, is working or not. Why? Because we are very much, because this is the first phase we are introducing our drug to the humans. So our priority is to check the safety of the patient or the participant. So we are taking the healthy individuals. Why we are taking the healthy individuals? Just to check that whether this drug is safe or not, whether the pharmacological aspects are being properly, what you can say, addressed or not. How I can say the pharmacological, you are seeing the point number B, like the characterization of drug absorption, distribution, metabolism, secretion. So these are the kind of things which we observe. So in a healthy individual, you can easily observe, right? And now your question will be like, why not we check it in the unhealthy individual? Because we don't know. Okay, and in healthy individual also, we are giving a very small dose to them, not the big dose. And this phase also lead to, you can see in point number 1, the maximum tolerated dose here. That means we are not introducing the drug into a one go. This is some kind, we are doing a type of design of the study will be like dose acceleration, like a little bit of dose you are giving, and then gradually you are seeing the dose, right? And when you reach that the maximum absorption has been done, or now you can't go beyond that, you will stop. So like this, you will go in this phase, right? So the additional objective of this phase is to evaluate the potential study endpoints, therapeutic regimen also. Okay, this is how you see the preliminary study of activities or the potential therapeutic benefit may be conducted in phase 1 as a secondary objective. So such studies are generally performed in late phases, but being appropriate when drug activity is readily measurable with a short duration of that exposure in the patient at early ages, right? Then we'll come to the phase 2. Okay, phase 2. This will be a I mean, like, this phase is also called as a therapeutic objectives. So the primary objective of this phase 2 is to evaluate the effectiveness. In case of drugs, in case of devices, we will evaluate the performance. Okay. Okay. One more point which I've missed in phase 1. Phase 1 trials can't be do in cancer patients and in for the cancer trial and for the medical trials, because you will not find a healthy individual you will expose with that medicine which has been meant for cancer, and you will not introduce that medical device to the healthy individual. So this is the kind of basically we can say the type of study design based upon for which therapeutic area we are working. So phase 1 is not applicable to such vulnerable kind of diseases or for the medical devices, right? So come to phase 2. Here the sample size a little bit increased, and here we determine the effectiveness, or we can say the performance of the of that particular drug or the device. And apart from this, because the time span is also very short for this phase, so short-term side effects and risk associated to it we also observe. What are the side effects? What is the technical term for side effect? That is adverse event, or the SAE, serious adverse event. So serious adverse event further lead to be like divided into further phases. Like serious, how you can say this is a serious? when the patient in the trial is having had a death, or after giving that drug, it is being like any kind of impairment happens to it. Suppose a ophthalmic drug I am giving and it leads to the visual impairment. Any kind of hospitalization, I'm inducing any kind of vaccine and it lead to the patient his what you can say, his oxygen level suddenly drops after giving that vaccine, so he'll land up into the ICU and it lead to the 2-3 days. Okay, so the prolonged hospitalization is another kind of SAE, you can say. And so these are the other what you can say, these are the various side effects, adverse events, and the serious adverse events. Okay, so in phase 2, it should conduct in a group of patients who are selected by narrow criteria. What is the narrow criteria? Narrow criteria here indicates as inclusion and exclusion criteria of that particular protocol or that particular study. Inclusion, here we are not taking the broader patients. We are just restricted to the naive patient of that particular disease or the particular indication. For example, if I am doing any trial, I want XYZ medicine and I have made it for wet AMD. However, you have seen in other researches or in the other publication or in the other papers, you have seen that this particular XYZ can be used for dry AMD and DME. So, but it doesn't mean that my project is clearly saying, my inclusion exclusion criteria are clearly saying is that we need a naive patient for wet AMD. That means only wet AMD. We don't need the other. So here the criteria goes narrows. Hence the sample size we put is usually very small, because we know that naive patient will be very the frequency will be very low, right? Why? Because in this current situation, patient might have other comorbidities. So the frequency or the percentage of getting that naive patient for that particular indication is very low, right? So that's why the sample size is very low, and we had a strict inclusion exclusion criteria for that. Okay, so these studies are being closely monitored. Monitored here means a proper observation is being there. Here I have highlighted the word monitor, which I later on in the upcoming slides I'll take you through that. So these objectives may be served by exploratory analysis, examining the subset data, and by including multiple endpoints in this, you can say, in this phase, right? Okay, apart from this, as an important goal for this phase, to determine the dose regimen for phase 3. Okay, because after the successful what you can say, completion of this trial may lead to the further phase 3 trial. So we have to also set a dose regimen. However, in this also the dose regimen is there, but what we can do is we keep it as low as possible. Okay, so the dose in phase 2 are usually lesser than the highest dose used in the phase 1. So the additional objective of this phase can evaluate the potential other study endpoints, the therapeutic regimen, the targeted population for the further studies. So these objectives may be served by exploratory analysis, as I said, and examining the subset data by including multiple endpoints in this trial. So moving forward to the phase 3, here the sample size little bit increased. However, there's no concession in inclusion exclusion criteria. Same inclusion exclusion criteria, little bit changes will be you can see, but inclusion exclusion criteria will be remain stringent in this as well. So why we call this phase as a confirmatory phase? Because here, by the time here, after the completion of this trial, we are having into that position, you know, the confirmatory position that yes, this particular drug or this particular active ingredient or the combination drug is working, right? So here it is to confirm the preliminary evidence accumulated in phase 2 that a drug is safe and effective for a use in intent indication and the recipient population. These studies should be intended to provide an adequate basis for marketing approval. Studies in phase 3 may also further explore the dose-response relationship use of the drug in wider population in a different stages of the disease, or a safety and efficacy of the drug in combination with the other drug, if the combination is there, in case the patient is suffering from any comorbid condition. If for drugs intended to be administered for long period, trial involves extended exposure to the drug. The studies carried out in phase 3 shall need to be completed the information needed to support adequate instruction for use of the drug. Example, the prescribing information that by that time because after this you are applying for regulatory approval, and regulatory for having okay my slide is moving or not, because I'm having one of the question. Nisha, can you confirm me that everybody you are on the same page? I'm like you all are seeing slide 6.
Yes, we're seeing slide 6, but sometimes the size of the screen is changing for some reason.
Okay. Okay, maybe the internet issues. Okay.
Maybe.
Maybe. Okay. Okay, for the prescribing information, suppose if you can't give this particular drug to those patients who are under hypertension, so such kind of information we need to gather in this phase, because you are applying for a regulatory approval for post-marketing, whereas in every phase regulatory approval is there because you need to submit it to the respective regulatory authority. But for marketing, you need the proper approval of this, the NOC kind of approval for this drug. So you should you should have the complete information, right? So going to the next phase of trial, that is post-marketing trial. So after being marketed, still you need to conduct a trial, because now you are performing this because this drug or whatever the vaccine, whatever it is being used by a wide population. So you need to check here the inclusion exclusion criteria are slightly different here, that are not that much stringent as compared to the earlier phases. Okay, so here especially regulatory authorities will say that you need to do, we have a what you can say a regulation for that, even for medical devices, because medical devices bypass phase 1 study, right? Or the other kind of trials, they bypass, who has a concession for phase 1, that they shouldn't have to perform that phase 1 study. So in that case, they have to perform phase 1. Even this phase is further classified as PMS, post-marketing surveillance, and PMCF, post-marketing clinical follow-up. You need to do that. The companies are bounded with that. They need to do that. So in that case, because how the medical device is being working, because we after inserting the device into the patient, you are having a very short time span. So how long? Because it's in the body for a very long period of time. So you need to check it. Okay. So because what happens, especially in medical device, after after having a surgery to implant that particular device into the body, patient usually don't come up when the bone fracture will happen and the plate or rod will be inserted, and after the bone union occurs, X-ray reports will come completely fine, patient happy, doctor happy. then patient usually don't come back to have a removal of that implant. They will be like, "Isse kuch problem nahi ho raha, to we'll move forward." Okay, so then don't prefer, but it might lead, because that corrosion or the erosion can happens after 15 or after 5 or 10 years, and such cases has been reported in UK. So after this, they have strictly said that post-marketing trials for everyone is mandatory. That's why drugs are performed after the approval of drug and related to the approval indication by the regulatory authorities. So let me tell you the famous regulatory authority's name. In US, you know that Food and Drug Administration is been working to give approval for drug, foods, and other any kind of things. So there are this authority is being provided their approval. Their further bifurcation as per department is there, but you can visit the website and you'll get to know about this. And in Japan, the Pharmaceutical and Medical Device Agency is there, who is regulating this. And in India, CDSO is regulating. I don't know how many of you knows this, because in India, you will go to that our regulatory department, you will see FDA Bhavan, the huge FDA Bhavan, and behind that a small CDSO office. So their CDSO office is there. The Central Drug Standard Control Organization, where the DCGI, the Drug Controller General of India, is giving approval in that. So Dr. Vijay Sumani is the current DCGI who gives the approval and sign on all these approvals. So this phase includes additional drug and drug interaction, dose response, safety studies, trial design to support use of the approval indication in mortality or morbidity studies, epidemiological studies, etc. Such trial go beyond the prior demonstration of drugs, safety, efficacy, and dose definitions. So, okay, these are all about the phases. So let's come about, we'll talk about the study design. So these are the various study design which we'll discuss, like a placebo-controlled trial design, active treatment, open label trial, parallel group trial, crossover design, excuse me, dose comparison, and the dose acceleration kind of studies, acceleration, or you can say de-acceleration studies. So first of all, we'll talk about the placebo. Placebo is a form of inert substance to stimulate a medical therapy without specificity of the condition being treated. So usually you will say that we haven't seen such kind of trial where the placebo is being given. So these kinds of trial are usually used in psychiatric trials where the patient is having some kind of medical condition. Where the patient is having some medical condition, they feel like this problem or this issue or particular psychological thing is being not treated. So what happened is like you are distributing two arms. You have made two arms in which you are giving one drug or another drug. So the other one will be a placebo, right? Just to avoid what you can say, just to because you have designed in that certain way, just to check that the normal people who are not in that treatment, whether this treatment is being worked or not. So in that case, psychiatric trial, usually this kind of placebo we use. Generally, if the standard of care is available in the market, is being already approved, you can't use placebo kind of trials. But if in case the standard of treatment is not there, you are doing it with some active drug, so then it is okay. So the placebo must share the same appearance, frequency, formulation as like an active drug. So if you are making a tablet, so that placebo should also resemble the same. So that if the patients are sitting in the OPD and they both are talking, I have received the red one and you have received the blue one, why it has been there. So both will exactly look like same. I remember in one of 6 years back, I've done one of the trial for non-infectious uveitis. And here we have a medical device in which that pharma company named Placebo has their device, a small size of the size will be less than the size of a rice, where they have inserted the active ingredient and they insert it inside the eye of the patient, where it will the active ingredient 0.02% will come from that particular device, and it will subsequently helps or in reducing non-infectious uveitis. So in that case, one of the group is getting that device, the another one is not getting. But in that case, what they have done, they made the same injection, okay, the sham one. However, they will treat the patient in the same way. They will take it to the OT, will do all the procedure and will have that injection, but the injection doesn't have that device. So this is how it will be like. And if suppose you are having one of the drug in the tablet form, another is in the syrup form, so what you will do, you will make another one into the tablet and one more syrup, so that both will look same. So this is how the placebo kind of studies goes on. So the placebo control helps to discriminate the outcome due to intervention or the new product from the outcome due to the other factors. The design is used to demonstrate the superiority or the equivalence. This design must be adopted only when no effective treatment exists, as I said. So placebo must be used with no permanent harm, any kind of that morbidity, or a cure by delaying available active treatment for the duration of trial, and is preferable for a minimal risk or short term, right? If the existing treatment is there, so you can't use placebo. Okay. So moving on to the open label trial, as the word indicates that open, here open means whatever you are giving it to the patient, both the patient also knows what I'm getting, and the investigator also knows what he is giving. So this is as the name indicates. So both investigator and the trial participants are fully aware of which treatment group the patients are in and what treatment are assigned to them. Open label trials are used to compare different treatments or gather information on the long-term effects of a new drug or treatment. And these trials can increase the confidence about the incidence rate. And the random allocation of the participants into the treatment on the control group is still necessary even in the open label trial to produce valuable data and pharmacological expected reaction to the drug and the treatment. But as they are typically biased and uncontrolled, this data is not for a great value to the researcher, right? Because they are generally these kinds of data have some kind of biasness. But you can't say that we can't do open label trial, because especially when it's come to the medical device, all medical devices are by default open label. Very rare it will be like the closed one, but in entire my journey to the clinical research, I've seen that all of them are like open label. Even the trials of cancer are also open label. If the standard therapy is there and the new active drug is there, so you need to disclose it to the patient, because sometimes regulatory body itself, when you submit your protocol to them or to the ethics committee, they suggest that you need to go for an open label because of the ethical consideration, right? So they will made you amend your protocol. So these are some examples of open label trial. Okay, then comes to the active treatment. Okay, comes to the active treatment here. This design involves the comparison of a new drug to the standard drug, or the compare combination of a new drug or the standard therapy, vice versa, the standard therapy alone also. Here you can see the in this picture is the participant group is there. You are screening the patients and you are giving it to the treatment. You are allocating them to the arms here. The red one is the treatment 1, treatment A. The green one is the treatment B, and the violet one is the treatment C. Here you are allocating like this. So why it is called active treatment? Because all the arms is getting some kind of active ingredients. Okay, it can be a standard of therapy. Suppose in treatment A you are giving the standard of therapy, which has been already marketed, and in treatment B you are giving the new active ingredient, or in treatment C you are giving the combination of both, right? So such thing happen. Yeah, can we do a surgical intervention to injection to inject a placebo ethically permissible? No, it is not. It is not ethically permittable to you, because as I said that if the placebo is not, it is an inert. So either you are giving any kind of injection or a drug, giving it as an oral. So it should, whatever form you are giving should come as it is. It will come out. So in case of injection also, if you are using any injection, so surgical intervention should not be happen, right? You can just put it in case of that particular trial, you can put it over the eye, you can make the patient feel yes that something has happened, but you can't go with any kind of dissection, or you can make any kind of incision or something. No, it is not ethically permittable. In fact, the ethics committee will raise a question, or either the DCGI, US FDA might raise the question, right? Okay. Yeah. So these treatments, as I said, will be there. So that's why it is called active treatment. Just give me a moment. Okay. So the therapeutic modality that should prefer being a current standard of care against which the active drug is being studied or is being compared with. This design can be used to demonstrate equivalence, non-inferiority, superiority kind of this study. I mean, like you are comparing a new drug, you are combining. So it can be anything, right? You are combining two different doses into one, and with the standard therapy, right? So, and you want to the objective of your study is just like treatment B is more therapeutic or more functional than treatment A, right? So these are kind of non-inferiority or superiority kind of studies. Or you can say sometimes it happens is like if both the drugs they are bioequivalent, right? So you can say that these both drugs are working well. However, the indication is same, but they are equivalent to another, right? Suppose ingredient A is used for treating you can say fever, and you have also a new substance B, and the same, it is being also used to treat fever, right? So here you can establish the equivalence. So the generic drugs usually go with this kind of study design, right? So here equivalence, non-inferiority, and superiority you can evaluate through this. This design is most effective and ethical. However, approved drugs and are available for disease under the studies. Okay. Okay, then I'll come over the crossover design. The crossover design here, as the word indicates, crossover. So somewhere into the line, it will they will cross the paths, right? So in this design, some participants start with drug A and then switch to drug B in one trial, while the subject in other arm will be in drug B and later on will be switched to drug A. So a certain method is there, not that they will randomly think that earlier doctor or the investigator is prescribing this one. So to someone else, he has prescribed this, so I will take this because doctor said that the crossover design will be there. No, because there is a certain time period and a frame when this crossover design will be happen. So how? You can see the red line over here and a green line. So these are the two parallel arms going on. So after screening, you have sent half of the patient to the red arm and half of the patient to green arm, giving them the red drug and the green drug. Over certain period of time, you are giving them the frequency, dose, formulation, everything will be same. Okay, you are giving them just to have it, will note down the symptoms, right? And after 6 months, after completion of the study, after 6 months from that startup of that dose, after exactly into the 6 months, what you will say, both the patients or both the treatment arms that you need to stop and give them the time period to wash. Okay, so approximately 1 or 2 months is being given. However, 1 week is enough, but it depends from drug to drug, what kind of drug is there and what is his absorption rate, what is his retention period into the body. So as per it, you have to give them that time. So if suppose I have given the 30 days, washout period will be 30 days. So after 30 days, then the subject with red drug will start getting the green drug, and the subject B who is getting the green drug will start his regimen with the red one. So these are the crossover design. So these designs are usually done if you are having some kind of what you can say, some rare disease and that particular indication you are finding difficulties. You know that the prevalence rate of this particular indication is very low, right? You will not find the patients, you'll find difficulty in the patient population. So generally this kinds of treatment or the design is being used by the investigators, doctors, or the pharmaceutical companies, right? So this kind of they will use that the same patients will be used. So whatever will be the output or whatever will be the data, they will record it as it is, and a particular computerized program is there to allocate them, right? So adequate washout period, as I said, is given before crossover to eliminate the effects or the initial administration intervention. So the requirement are as twofold. The disease must be chronic, stable, incurable. Characteristics must not vary for the duration of 2 study periods and interim wash period. Bioequivalence and the biosimilar equivalence studies usually utilize a crossover design. The duration of follow-up for the patient is longer than the parallel design, and there is a risk that significant number of patients do not complete the study and drop out, which lead to the compromise of the study power. So yeah, this is one of the reason. However, these are the reason which sometimes leads to you are not get the desired sample size, because dropout rate will be there. But this is not a limitation of this particular design. This is the limitation of every design. Whatever you will follow, you have to follow some kind of patient retention kind of policy, or you need to repetitively counsel the patient that this is very much important. You don't need to leave the study because this particular disease is will if you will leave the treatment, it will be lead to more lead you to the more problematic situations, right? So like this, you need to regularly consent. You need to regularly be in touch with the patient. So dropout is not the only reason, or you can say the limitation of the study. It is the limitation for every design. Okay, then we'll come to the dose comparison and dose acceleration studies. So here we can compare different dose, what you can say, the regimen, not different dose, regimen you can say, into the same study. Suppose in one of the treatment group, you are giving one of the medicine with 50 mg, another group with the 30 mg, right? And another with the 10 mg, just to check that which is more effective, or either all three are effective. So this is dose comparison. And it sometimes also club up with the dose acceleration kind of studies, right? With the same patient, within the same patient, not the different treatment, within the same patient, you are giving, suppose, the 5 mg of drug, and then later on you will accelerate that dose into the 10 mg. Okay, then you will see how that patient's body usually responds. And these kind of drug usually are made up into the cancerous, because where what we are doing, we are measuring the body mass index of the patient, and based upon that we are calculating the dose. So such kind of studies being done in that cancerous patients also. So such acceleration and deacceleration, vice versa, also happens. Depends upon the indication, the active ingredients, how it is working, right? So such things happen. So the different dose and the regimen of the same treatment are used as an active arm and in the control in this design. The purpose is to establish a relationship between dose and efficacy, safety of the treatment. This design may be insufficient if the therapeutic range of the drug is unknown. Yes, if especially for the new active ingredient, if you don't know the therapeutic range, when it is being started, you can observe that yes, after this dose, you can see a difference in the patient. So if that doesn't know, so you can't proceed with this study. But usually now we have various uh, what you can say, various methods by which we have done such development still, that we can analyze this thing, that what will be the therapeutic range of that drug. So this also include granular increase and decrease of dose regimen, as I earlier said, especially in the acceleration and the deacceleration kind of studies. Okay, so these are the designs which I have talked. There are few more things which I'll when I'll take you through the methods to reduce biasness, then I'll club those things and I'll show you a broader picture of a trial design. So while designing a trial, or you can say a research, so what are the factors which you need to keep in mind? So the number and the characterization of treatment to be compared. Okay, characterization of the disease under study, the study objectives, this matters, on which phase you are doing and what is your objective. However, the primary objective never changes, like safety, efficacy remains the primary objective. The secondary objective will be like as per your what you are looking for, right? And the same as depending upon the objective, the primary and the secondary endpoints changes. Time frame, because okay, time frame, it matters, because the longer duration you will set up for your design, so the dropout rate will be there, the withdrawal will be there. So through that you will lose your data, right? Whatever you are expecting to be have at the time of statistical analysis, you will not get that sufficient amount of data through which you need to put your results, right? Then comes to the treatment course and duration. As I said, carryover effects, duration of the study, which is linked with the dropout rate. As I said, these all are correlated with one another. The cost and logistic, this is the most important thing. Then patient convenience, yeah, you have put a schedule of visits, and there are a lot of visits in the study, that every suppose every 2 days or every 4 days the patient has to come. So this shouldn't be there, because patient will come, and after a certain time they will be like, okay, we are not convenient to come because you are frequently calling us, and we went to that doctor, he said that he will treat me, and within after 3 visits I have been treated well. So this happens one of our study, in which 13 doses of the ranibizumab has been administered to the patients over. So the follow-up is of 1 year. So what happened is, like, after giving 1 or 2 or 3 doses of that drug, eventually the patient vision will come back. So they wouldn't come, right? One of the factors is this. And after seeing that, oh my God, for one whole year we need to come. So here patient convenience is there, but you need to regularly had been in touch with the patient. So whenever he will visit, you need to know about the importance of being in the trial, or importance of his how critical is his condition. So if he will drop out, he wouldn't be no longer the participant of the studies. You need to make the patient aware of risk as well as the benefit of the studies. Apart from this, you need to see the ethical consideration. You shouldn't make such kind of design which lead to raise a question on the ethical aspects. Then comes to the statistical consideration. Yes, your sample size plays a huge amount. So whenever you are thinking that I am designing a trial and I'm putting 100 of the patient, so just please take a note of the expected withdrawal, the expected dropout, or the data missing. Because what happens if suppose I'm a patient and you have a study, so I'll come for visit 1, 2, 3, and for the 4th visit I haven't come due to some mishappening, or certain I have to attend some marriage or a function. I went there, I didn't come for visit 4, and then I come for 5, 6. So this data, the missing data, it's create a problem while doing the calculation for your study. So what you will do, you will eventually eliminate. However, the patient is regular, but for one particular visit he didn't come. So in that case, you need to you need to keep in mind. So the sample size will be like, if I know that if the pure 50 patients will be there in my study, my data will be reflect in that way, which I have think my objective will be completed, or will eventually get that result which I want, or which I am expecting, right? But you have to take 100 patients, because you need the withdrawal. Because patient will after coming, madam, we are moving out of the state, we can't move ahead with this trial. So they will take withdrawal. Other one, as I said, the missing data and the dropout. Some will be like the non-compliance one. They will say, yes, I will come, definitely. But eventually, after some time, they wouldn't come. You will call them. I've seen that so many doctors used to call their patients, that's just please come, especially in the trials. And patients sometimes don't care, they wouldn't come. They will go somewhere else, right? It's not like they will not come or they will go somewhere else. So these things you have to consider. Then study subject availability, the disease rarity. I mean, like you have to also sees that which indications you are working for. Rare disease, it has been very difficult for the doctors and for the site staff, because the frequency and the prevalence rate is very low. So here you need to think on the aspects of trial design and to the sample size also. Then comes to the inter and intra subject variability, and this is the subsequent part. Okay, so these are the factors which affects the trial design selections. Okay, then we'll come to the biasness. You know that biasness, you all know the definition. The chance errors which lead to the deviation from the result, which can be done intentionally or unintentionally. Yes. Sometimes we do, if we have some personal interest to the particular study, so we do it for intentional, and sometimes it happens unintentionally. The errors happens unintentionally. For example, how I can explain you. Okay. One of the example I'll give you of selection bias. Okay. Usually there are various type of biasness. Example: selection biasness, measurement biasness, analysis biasness, publication biasness, or the reader biasness, you can say, or the funding biasness, or right. So the selection biasness will be like selection and allocating of the patients. So giving one example, in the HIV trials, you will see that male population will be higher than the female population. It is not that there is no access data that the prevalence rate in the male for HIV is higher than female. No, they are equally both. That this particular HIV will the rate of prevalence is equally in both, right? Because it is not any kind of genetic. So why it happens? Because usually female are very much hesitant, so they don't come up for the trial, right? So what you will see that majorly male population is there. So this is an unintentional. You don't want it intentionally. So it is an unintentional that only male patient you will get, right? Measurement biasness will be like regarding to the instrument. Suppose, right? If you are using a certain device to check a certain parameter. So earlier you have done with device A, and then later on you are going to device B, C. So because it might happen that device B, C are not calibrated, so your parameters will be fluctuating. So this can be happen. Then analytic biasness, dropout rate, loss of follow-up. This happens is when usually you will see that lot of AEs has been happened, and the serious adverse event has been happened. So you are suggesting patient just to drop out from the study. Another is like funding. Funding here, it totally depends upon the sponsor, because if suppose I am the sponsor and I am, apart from the trial, I am sponsoring for various programs which you are conducting at your facility. So you will be like more what you can say, loyal, or to be more diverted towards me. So you want to be in that way, right? So such biasness usually happens. So there are different ways to overcome these biasness, which I'll later talk. So come up to the methods of reducing biasness. So these are the methods of reducing biasness: randomization in clinical trial, blinding, stratification, monitoring, inspection, and audit. So these are the different method. So come to the randomized control trial. Now, earlier you have read all those trial design, how we can make, and we are saying that you can allocate the patient to any of the arm as per your choice, or you can say as per the trial design. So the allocation should not be depend upon you, because here you can do any kind of selection biasness. Suppose, oh, I know the patient, this is the active treatment, the standard active treatment is more effective than the standard one, so I'll keep him in the active treatment, not in the standard treatment, right? So the randomized control study will be like randomly you are assigning it to the patient, and this random control, how you will do, you will remove it through a centralized process or introducing an electronic randomization kind of system. So what are the electronic randomization systems we are nowadays we are using? We are using IWRS, interactive voice response system, or interactive web response system. Sorry, the same has been here. I need to change it to the web. You just consider it to be as a web. Both the things are same. Here we are, whatever the characteristics we are taking of that patient, suppose the patient age, height, weight, or at what baseline he is been there, right? So we are filling that particular baseline data to the system. In case of web, you have that particular website which is being allotted by either the sponsor or that person who is interested in that trial, who has designed this trial. So that person will give you that centralized link for that randomization. Through that, you need to just put on the parameters, the baseline parameters, and the system will randomly give you that yes, active treatment should be given to the patient, or the treatment B should be given to the patient. And in the interactive voice response system, what will happen is like through the mobile phone, a simple toll-free number is being given where you need to type A, B options, and based upon that, you will get to know yes, whether you need to allocate this patient to the treatment A or to the treatment B, like this. Okay. So this randomization, earlier how randomization will be done, people used to done, is it in the Excel sheet? If I have to enroll 100 subjects, I'll write the 100 enrollment number and allocate it 1:1. Treatment 1, treatment 1, treatment 2, treatment 1, treatment 2, like this. Okay. So once this might happens like that error, the chances of error is being there, or the chances of biasness is being there. So it will eventually lead to those outcome which you don't want, or you want, in either of the way, right? So the process of randomly assigning a trial participant to a treatment or to the control arm is called randomization. Randomization removes potentials for systematic errors or biasness. The central randomization is preferred, and these are the following. Central randomization, how? Now, if you are institute based, you are doing a study within an institute, then this centralized randomization wouldn't be there for you. But if a pharmaceutical company is doing a research, suppose in India, and in India there are 50 sites, so you can see a centralized randomization will be there. Site 1 has enrolled 1 subject today, and site 2 has also enrolled. So whosoever has enrolled, feed the details in the system earlier, will be assigned as 001 number, and in site 2 will be assigned as 002. So your next, at site 1, your next patient will be like 004. The enrollment number will be eventually come out with a treatment. So it might happen, there are chances that all your patient in that particular site will be on the active ingredient or on the sham ingredient. This might be happen. This is the possibility. Why? Because of centralized randomization. In that case, you don't think that sponsor is only giving us sham, because in one of my case, this happened when I was working with one of the study, another hospital, Dr. Shroff Hospital. Here, I have observed that the investigator was saying that, Pooja, all the patients are under sham. So is there any error? You just please confirm it with the sponsor. And after I have confirmed with the sponsor that there is no error, they have done a centralized randomization. This I have to tell her that this is a centralized randomization. So the chances are like 50-50. So it happens that your all patient is receiving sham drug or the sham injection. Okay, this is one of the how to reduce the type, how to reduce. Another type is blinding. As you know that blinding, when it's come to blinding, you have to blindfold yourself. So it is opposite to the study design called open trial. Now it is a closed trial. You can say closed trial also, blinding also, and masking also. In open trial, everybody knows which drug is being given, which treatment is given, in which arm I am. As I said that in the medical device one, bone fracture is there, the femur bone fracture is there. So half of the patient you are putting plate and half of the patient you are putting rods, right? So both the patient knows that I have received a plate and he has received a rod. So this is an open. In the blind one, neither patient knows, but that the blinded one will not be in case of medical device. I'll talk about in drugs, that both the patient will be none of them or any of them will know which treatment you have been allocated, just to avoid biasness. Because yes, it might happen that you know for which treatment you have given, so you will treat the patient according into that way. So in an unblinded or open study, the information about the assigned treatment is available to all the participants and group involved in the research, right? Blinding or masking is the process by which information that has the potential to influence the study result is withheld from one or more parties involved in the research and studies. So if giving you one example, if in case two different sponsor of pharmaceutical companies drug is there and they are bioequivalent, right? Both are bioequivalent. But you know that one of the sponsor is very much professionally been associated with you with such a long time, and they have introduced this study. So you might be get biased toward that sponsor, right? However, the medicine or the drug is same, the equivalence is same, everything is same, the frequency, dose, regimen, everything is same. But yes, you can be biased, right? Because for my institute, this every program, everything is being sponsored by, example, the IQVIA has sponsored everything. So this is like this, okay? So this further blinding is further divided into 3 parts, right? You can do a single blind study, double blind study, or a triple blind study. Okay, so let me take you through this. Single blind study here will be like the patient doesn't know which treatment is being given. This will be the single blind, right? Rest of the stakeholders will know, like investigator will know which treatment I'm giving to this patient and which treatment I'm giving to that patient, right? Sponsor also know everything, who are into that trial, everybody knows except the patient, the end user, you can say. Then comes to the double blind. Here, neither the doctor knows which treatment I'm giving, nor the patient knows which treatment I'm receiving. But the sponsor, the person who's conducting the research knows which treatment is being allocated to which patients, right? And then comes to the triple blind. Triple blind will be like neither patient knows, nor doctor knows, and even the sponsor doesn't know which treatment is giving. Here, usually sponsor hire a third party and they give the randomization and blinded related all details to him, that look, you are the person, you do whatever you want to do. These are my ratios. I want drug to be in, if 3 arms is there, 1:2:1 or 1:1:1. I am deciding the ratio for you, right? And you just randomly allocate it. So in that case, none of the party knows which drug is being given. So if in rare condition, if something happens to the patient and doctors wants to know which treatment is given, to that patient because his condition is very much critical. And after knowing that which drug is being given, based on that judgment, I will initiate the rescue therapy. In that case, what will happen? You need to request it to the sponsor, and sponsor will request it to the third party. They will disclose it. They will disclose which treatment is given. But for that particular thing, you will not consider it into your data analysis. However, you have to notify it to the DCGI, the CDSCO, health regulatory authorities, and even to the ethics committee also. Because why you have done? They have given you approval on that blinded study that you will not disclose, but what is the requirement? So the proper justification should be there. What is the rationale behind you have requested for this? However, you will, whatever the mail you will write, the sponsor will eventually look and will see, evaluate, because their own medical team is also there. So such things will be there. So these are the three types of blinding which is being preferred according to the design. So now you can design your own trial, like randomize, a multi-center randomized blind trial for this and this indication with the so and so drug, right? This is how you will design your trial. Then comes to stratified. Okay, now sometimes this is one of the factors which will reduce your biasness, because you are completely allocating half of the patient to this and half of the patient to this, and this whole will go long till the study will get closed. So some biasness, as I said, that they are not intentionally, they are unintentional, right? That might happens, and for that none of the person knows that what kind of biasness can be done. So to avoid such biasness, we are doing stratification. Here in the picture you can depict it, half of them are the patient pool is being allocated into the blue one, and half of the one are into the olive green kind of, and then further bifurcation is there, right? Further bifurcation is there. So I'll give you one of the examples. Just let me read the slide first. The objective of the stratified randomization is to ensure balance of the treatment group with respect to the various combination of the prognostic variables. This method can be used to achieve balance among group in terms of subject characteristics, covariance. So stratas are designed to avoid confounding factors. So now I'll give you one of the example. What are the confounding factors? In one of the study, what we have observed that we were doing the study for wet AMD, right? Purely the naive patients of wet AMD. And the prevalence rate of that wet AMD is higher in light iris rather than in dark iris. So what will happen? And the light iris frequency is slightly low as compared to the dark iris in the world. So it might happen that your all the patients are like with the dark iris, okay? And it might happen that the recovery rate of dark iris is quite good as compared to light iris, right? So what will happen? It will somehow affect your outcome, which you don't know, right? So how you will overcome this? You will overcome like through randomized. First of all, you have randomized that a treatment group A and treatment group B. Okay. Now you will further randomize those things, those patients, those groups into subset. Like if 50 patients are there, then I further divide it into the light iris group and the dark iris group, and here the light iris group and the dark iris group. Suppose for this study wet AMD I'm talking about, I have recruited patients with the vision BCVA score of 34 to 64, and I've kept 50 as a medium baseline. So I'll say that from 34 to 54 will come to one strata. So from 55 to 64 will come to another strata, right? And from this strata, I will give treatment to half of them to the treatment A and half to the treatment B. Same with this, half treatment A, half treatment B. And then further, I will classify, subset them as a light iris or dark iris. Here you will completely remove the biasness. It is one of the cases that you will avoid confounding factors, okay, by stratifying them. So your study, if you are designing a study, it will be like multicentric, global, randomized, blinded, stratified clinical trial for wet AMD with the treatment of XYZ, whatever. Okay, comes to the next, the overview, which earlier I have talked about monitoring. So apart from this, to avoid biasness, monitoring, inspection, audit usually happen in the clinical trial. So what will happen in monitoring case and in inspection case? What will happen? A delegated person or a third party hire will be there from the sponsor or the person who is financing that research, right? Will become and will see that whatever actual data we are getting on papers in the OPD sheets is being properly transcribed to the eCRF, the electronic CRF, the case report, because whatever you are doing, you are electronically transcribing it, and the back end, your biostatistician is regularly checking it, that yes, how the graph goes on for BCVA. If you are looking for a vision, that how it goes on. If you want to see that how much edema is being reduced after giving that drug at the posterior part of the eye, the macular region, how much edema is being reduced, is it being reduced or not, right? So that overview is there. Inspection and audits, subsequent audits will be there. Audits and inspection are usually done to check that ethical aspects of the trial is being done or not. Suppose you have recruited patient without consenting. You have just you know that naive patient is required for this treatment. However, the patient is having some other kind of comorbidity. Even you have written everything in your OPD sheets in the medical history. However, you have not mentioned it in your ECRF because you know that this is not our criteria. So eventually, it will lead to the eventually it will affect your outcomes. Okay, either in the positive way or in the negative way. So this is the overview. So it is an act of overseeing the progress of clinical trial and to ensure that it is being conducted, recorded, and reported in accordance with the protocol, SOP, GCP, or the regulatory requirement which is being required. All right. So these are the references, and thank you. That's all from my side. Any questions?
Hi, thank you so much for the presentation. It was a very detailed presentation. So can you please switch on to the slide which had placebo? You mentioned about the study, you injected a placebo in the non-infectious uveitis.
Yes. Yes. Yeah. Can you see the slide? Yeah. Yes, madam. What's your question? You need to know about placebo kind of trial?
Yeah, so I wanted to know, you told me about a study in which you injected a placebo in a non-infectious uveitis.
Okay.
So where was the placebo injected?
Placebo, that was a device injection in which one of the devices there that which I've talked about. Another was the blank. There's no device. The small device was not inserted into that. Right, I am not remembering the actual active ingredient name.
No.
I think
So
triamcinolone.
my question is, was it given? Was it implanted in the vitreous cavity? Was the injection given inside the eye?
Yeah, inside the eye. Yes.
Yeah. So that was
Yeah.
the question by Dr. Devipriya. That is it ethical to give an intravitreal injection with something which is not medicated? We are entering inside the eye. We are doing an invasive procedure. We are breaching the continuity of the eye. So is it ethical to do something with the placebo? Because we are not giving any, we are risking the surgical risk of a surgical procedure, but we are not giving any medication. So is it ethical?
No, it is not ethical. Let me clear you. In that, the injection which is having the actual device, right? The actual device is being surgically inserted inside the eye.
Okay.
Okay. Whereas, can you hear my voice?
Yes,
Okay.
I can hear.
Whereas in the subsequent one, the another one, the sham is being just placed over the what you can say, the cornea region. It will just give you a sensation that something is there. The pinch will be there, just like something is being touched to your skin. That patient will feel, yeah, I have been injected. However, no surgical intervention is there.
Got it. Yes. Okay. Thank you so much for clarifying.
If they want to read more about that, you can go to the website of PC Diva.
Okay, can you share the write in the chat box?
I'll write it in the chat box so that you can go to the website. This is the sponsor actually. PC Diva. So, and that studies of 3 years, because it is the drug is being been illuminated from that device 0.2% per day. So you can imagine that you are bypassing the those condition of corticosteroids, which is being given like 60 mg, then 50, and you will continually go moving ahead with the tapering dose. So you know that these drugs are how much harmful they are. They are continuously being piled up into the body. So they have that was a quite good you can say invented a good device, but I don't know whether it is being marketed, because that time it was under phase 2 6 years back. So 3-year studies was there. So you need to check that what is the current status. Even I will also check what is the current status of that.
Okay, thank you so much for clarifying. Do we have any other queries? If the participants have any queries, they can raise their hand, they can unmute themselves and they can ask.
So very good lecture by Ms. Pooja. So thank you so much. I just want to know if the clinical trial companies and all, they come over and they ask you to intervene and place certain devices inside. What is the legal protection, or what are the medical legal aspects of this? Like, can we as physicians or surgeons, can we go ahead without knowing what are the interactions of the drug? Like, how are we placed if we are requested to place such devices?
First of all, none of the company will directly approach you, because before approaching you, they need to take approvals. As I said, from every phase, they need to go to the regulatory bodies, and they cannot initiate a single step in the site until unless they has not been get a particular NOC for every phase, right? And then if suppose they bypass that, then your own institutional ethics committee is there to evaluate that thing. And after apart from this, if you are an investigator and some company is been approaching you, you have the right to ask each and every data of that particular study before initiating or before signing any contract.
Okay, and if some untoward complication or something happens and there is some case registered, so how are we protected against it?
The investigator you are talking about, or in the behalf of patient you want to talk about protection?
As an investigator.
As an investigator, because see, risk will be there, but eventually that has been already declared by sponsor that whenever some mishappening happens, so root cause analysis will be there. So usually it doesn't happen because in lot of uh, patient pool and with a lot of multi-centric trial, it might happen that you are encountering that case for the first time. So they will not uh, uh, make you stand in a court or into the legal things. They will check throughout the trial, they will check. And if the same case is being reported somewhere else, so it might happen that there is a functional error in that device or in that drug. That, or sometimes it happens like the other factors also being involved in this, because so many other investigators are there, and so many other instruments we are using if it has been there. So it might happens that, so it will not directly come to the investigator. It usually happens where very rare. In that case, if falsification data is being there or plagiarized data you are giving, in that case, usually the investigator is very much responsible. And rest of the cases not, because because you are doing the trial on that active ingredient or on that new medical device, which you don't know how it will act in human beings. So during trial phase, investigators are not that much, they will not make you stand in that way. And regarding study participants, whosoever is participating in the study, they company will do a group insurance for every country, for every participants. So whatever, even the essay, even the death will happen if in case. So the insurance is there. Insurance is there, and based upon that insurance, the company will bear each and every expense. And for that insurance, we have to declare that insurance to ethics also and to the regulatory body also, because insurance is as critical as protocol design. So until unless you don't have that policy, they will not approve it. So from both the ends, it has been secured. Very less cases there, as I said, the fabrication and falsification, plagiarized kind of data. If you are submitting, you are doing some intention kind of errors. So in that case, if you are found to be guilty, in that case, they will raise a question on your integrity. Rest, it usually doesn't happen. In entire my case, it doesn't happen.
Thank you so much.
Okay.
Dr. Nisha, I had a question also. This is Dr.
Yeah.
Yeah, so I just wanted to know, Madam was talking about the different phases, phase 1, phase 2, etc. So before starting these studies or trials on human beings, is it necessary that we have animal experiments, or it can straight away be started on humans, or how is it? Is it that some drugs have to be tested on for the pharmacokinetics, pharmacodynamics, other effects? How is it done? Like where are animal experiments a part of the trial?
Okay. If you have found some new entity, the new drug entity, in that case, it is very much mandatory to go through preclinical trials and the cell testing trials, just to see cell toxicity, is there any buildup, or is that particular drug entity is responsible for any indication which lead to further any kind of carcinogenic effect inside the body. And furthermore, the animal testings will be there, and the animals are always selected in that certain way. For example, in case of eyes, we are taking, if you want to inject something like Ozodox or the injection which I have said, that in that case you need a pig eye, because pig eye cornea will resemble to the human eye cornea, right? And if in case you are testing any drops, so rabbit eye will be there, because it is as sensitive as human eye. So this is how they will select the animals. And if the particular entity or the drug is being already marketized, suppose X ingredient is being already used, for example for wet AMD, now you want to explore it into the dry AMD or in DME cases also. So you know the safety of, you already know the safety of that particular substance, a particular entity. So you don't require, in that case, you don't require, because you have the previous data. You will do meta-analysis and the research publication to establish the safety and the tolerability. However, you need to do phase 1, phase 2, phase 3, because it might happen that in wet AMD, 0.2% you are giving, and for dry AMD, it requires slightly less or higher to this. So just to set the dose for other indication, you need to go through phase 1, phase 2, phase 3. So in that case, you don't require the animal testing or cell testing. But if the entity is completely new, you doesn't know anything, you have to go through these parameters. In fact, you need to also go through CDD, computer-based drug designing. In one of my study in oncology, I have seen they have few what you can say the active ingredients, and they were checking the binding efficacy of these 4 active ingredients with the gene, the protein, that is HER2. So because if who has the better compatibility to join with that gene virtually, they are doing it in the system based. That process is called docking. You all know that if the binding affinity is more high in any of that will be considered as the best one out of 4. The ranking will be depends upon the docking. So advanced trials is being also simultaneously being going on. One point I also want to add up here, in advanced clinical trial, one more thing is there, because if suppose 1 year duration is there, you have set a 1 year follow period to the trial, and you don't know what will be the outcome, whether the outcome, the result will match as per your endpoints or not. Okay, you want to see, and that particular active ingredient, you are not aware about how it will work in phase 3 or in phase 2. So what you will do, you can do interim analysis also. What interim analysis is, after 6 months from the last patient has recruited, after 6 months, you collect whatever data you get, drop out or data missing, whatever, you just get do the calculation, just check. It will give you a slight projection of your end data, that how it will go. Because sometimes what happens is, like, you will project, you will see how the graph is going, whether it is going upward or downward, or it is going upward with so many adverse effects or the essays. So you will get an idea, because you are putting a lot of human resources, cost, everything. So here you will get an idea whether you need to terminate the study or you need to go further with the study. So this is one of the advanced way where they are doing interim analysis in between the data, between the trial. After, in the midway, they are analyzing whether they are going into the right path or not. If they are feeling like they are not achieving their own what you can say the objective, they can slightly modify it. Here the protocol amendment also come. Okay, you know that this is being not going into the right way. If I'll remove this particular inclusion criteria or exclusion criteria or the parameter, so the things will be like more set up, right? Suppose for any drug you have given, you have said that edema will go down, and eventually you have in the secondary objective, you are in the endpoint, you have seen that anterior chamber cells will also eventually reduce, but your secondary endpoint is not meeting when you have done the interim analysis. So what you can do, you can make an amendment. You can say that I will remove this secondary objective. We will keep the primary objective as it is, my primary objective or the primary endpoint, and will declare regulatory authorities as it is, like, yeah, previously it happens and during interim analysis we found this, so we will suggest that it will only work on the macular region or the edema, just to reduce the swelling of the macular region. So like this you can do. These are the advanced form of clinical research which you can introduce. So regarding that animal testing, your question is being well answered or not? I'm like, you
Yes,
understood?
thank you. Thank you.
Okay. Any other question? I have written my email ID also. If somebody is having a question, so you can direct it to this email ID, or you can take my contact number from Dr. Nisha. You can WhatsApp me. And I'll share this PPT to Dr. Nisha, and she will forward it to you if you need.
Yeah.
You can also directly contact her.
So I think we are already above the time. So, and if we don't have any other queries, we can end the session. So thank you so much, Pooja, for taking this lecture, and thank you all the participants for attending. I just want to tell everyone that this was the last lecture of our series, and we had a Shark Tank lined up, but till date we have got only 2 entries from one participant only. So I request everyone to please send their entries if you have any research idea, but that will happen only maybe in the 2nd or 3rd week of November, because next weekend we have the annual conference of Women Ophthalmic Society. So there won't be any, even if we have more entries, the session won't be there in the next weekend. So I hope to see most of you there in the conference, and thank you so much everyone who took out time on the Sundays for last 3 months. And I think we should end the session now. And regarding the recordings, we'll share, because our the support team, the IT support team, is very busy with the conference. So maybe in the second, by the end of the November, we'll share the link for all the recorded videos, and we'll send out the participation certificates also.
Thank you, Dr. Nisha. You've
Thank you,
been
ma'am.
great, and it was a great presentation and hosting also on all the Sundays.
Thank you so much.
To you. Yeah, you did it very well.
Thank you, ma'am.
So the certificates will be mailed to us?
Yes, ma'am, we'll mail. I talked to Mohita ma'am also, but I think we'll mail.
Thank you so much.
Yes, thank you, ma'am.